Good afternoon and welcome to another Research in Action brought to you by the Division of Research at Florida Atlantic University. We're very glad you're here. Just as a reminder, this is a weekly event. Every Thursday at 1 p.m. we discuss very different topics. You can find those listed on our website, Research in Action at FAU. Today we're going to talk about cancer. Before we go there, we would very much like your questions and address your questions, but because of the number of attendees that we're having, we do not allow verbal audio questions. So if you have your mouse in the bottom of your screen, there's a Q&A button there. If you push that Q&A button, a little pop-up, a window will come up, and you can type your question there. You can type your question at any given time during the presentation. We will address as many questions as we can after the presentation, and those questions that we won't have time for, we will ask our presenter to answer offline, and then put those on our website together with a recording of this presentation. With that, it is my great pleasure to introduce to you Dr. Gregg Fields, today's speaker. Dr. Fields is professor in chemistry and biochemistry in the Charles E. Schmidt College of Science at Florida Atlantic University, and he's also the executive director of the Institute for Human Health and Disease Intervention, iHealth for short, which is one of our, what we call a pillar at FAU, one of our research institutes. Dr. Fields also holds an adjunct professor appointment in the Department of Chemistry at the Scripps Florida Institute in Jupiter. Dr. Fields has very many years of experience in the field of extra matrix proteins and has been at a number of different institutions before coming to FAU. He holds various awards. I'm not going to go into those, and it also has been inducted in the National Academy of Inventors fairly recently. With that, I'm just going to turn it over to Dr. Fields to tell us about new perspectives in cancer treatment.
Okay. Well, thank you very much, Karen. I appreciate that introduction. Okay, so what I'm going to talk about today are some relatively new cancer treatments that come along probably in about the last 5 or so years, and some ideas about where these treatments are going, some of their limitations, as well as, the success stories also. So what I want to talk about first is really the long road in battling cancer. So this is an ancient disease, okay? I mean, here we have a picture of a medieval man where there was a tumor present in their leg. Cancer has been known about for a long, long time. Now, here in the United States, we really started a concerted effort to look at cancer about 100 years ago. First, there was a formation of the American Association for Cancer Research, which is just really brought together a number of scientists who were devoted to doing cancer-based research. Then in 1913, we had the creation of the American Cancer Society. That was the first really substantial foundation which was going to raise money for cancer research. In 1937 was the creation of the National Cancer Institute within the National Institutes of Health. So that was a federal initiative that brought together cancer researchers in one building, as well as providing a support mechanism for people doing cancer research throughout the U.S. Then in 1971, we had the National Cancer Act, and this provided additional money for cancer research, but also created the Fredericks Cancer Research and Development Center in Fort Detrick. So Fort Detrick, unfortunately, was actually a place where they were developing bioweapons, and they did walk away from that and then turn it into a cancer research center. And that deals mainly with chemotherapy. Then in 1993, we had the National Comprehensive Cancer Network. So this involves various institutes across the United States that have been deemed comprehensive cancer centers. And they now have a network where they interact with each other. Here in Florida, we have one comprehensive cancer center, and that's the one that's in Moffett. Then in 2015 was the creation of what was initially called Cancer Moonshot 2020, or now it's called Cancer Breakthroughs 2020, and this dealt with looking at immunotherapy approaches for cancer treatment. And then also I'll do a quick shout out to the state of Florida, where we use some of the tobacco money that we got to battle cancer, and they created the Florida Department of Health Cancer Centers of Excellence, and that was in 2015. So let's start with the pillars of cancer therapy. What are the different ways that we treat cancer? And some of these, I presume, are going to be fairly well known, some maybe not so much. Well, surgery is often the first option, depending upon when cancer is detected. Radiation treatment, that's certainly been around for a long time, and chemotherapy. These are the ones that I presume most are familiar with. These are the ones we've been working with cancer for a long time. Now we've had approaches that deal more with targeting cancer based on specific behaviors of cancer cells, and I'll talk a bit about that. And then we have immunotherapy, which has probably been the biggest breakthrough over the last decade in terms of cancer treatment. Now, what's been very important in cancer treatment has actually been cancer detection, because I'm sure many are aware that the sooner you can detect cancer, the easier it is to treat. So there have been quite a number of what we call imaging advances over the years. And as we get better at cancer detection and determining what stage it's at, we can, first of all, do diagnoses at earlier stages. We get a much better picture of how much the disease has spread. Obviously, this helps patients receive their optimal treatment. And really what we want is to have more limited, minimally invasive surgical techniques. We want a good quality of life after these treatments. And then obviously, once the treatment has occurred, we can do a response assessment. And that tells us whether we need to continue the treatment or discontinue it. Now, along the lines of these imaging advances include these digital mammograms, PET, CT, and MRI. So this has really helped us in our fight against cancer because it's allowed us to detect cancer at a much earlier stage. And really, we have seen successes. I know at times it seems like, almost everyone is affected by cancer within their family or friends. But nonetheless, if we look at the cancer mortality rate per 100,000, you can see this has steadily declined over a reasonable period of time since the 1990s. The number of cancer survivors has certainly increased. So we are doing some things well, and there's certain cancers that are relatively treatable these days. Others are a little bit more difficult, and we'll talk about that. Now, these are just some of the important steps in what we call cancer genomics. So really what's helped us in treating cancer is what we call the beginning of the precision medicine era. And that's allowed us to look at the biology of cancer in terms of the different genes that are increased or decreased, different mutations that occur. So this started in about 2000 when the hallmarks of cancer were published. Then in 2005 is the Cancer Genome Atlas, which really allows researchers to look at all types of cancers, different examples from patients, what type of genes have been overexpressed, underexpressed, or mutated. And then we get into genetic subtyping of cancers, such as glioblastoma, gastric cancers, and breast cancers, and so on. And that's been identified through this TCGA that was launched just really about 5 years earlier. And this has become very important because it's allowed us to understand cancer on a much more molecular level. And what I mean by that is understanding the genes that are important for cancer to progress, understanding if there are different genes that are important in different cancers, because this can get very complicated, but really dictates the type of treatment strategy that may work. So this next slide there's a lot of information here, but I think this is a very, very important slide, because one of the things that often comes up is, why isn't there a cure for cancer? And as we look at the data for different cancers, I think we can understand why there will never be one cure for cancer, because cancer is really a group of diseases. So on this axis are different cancers, okay? So we have things here as breast cancer, brain cancer, leukemia, and so on. On this axis, these are just different genes that are often mutated in cancer. So what I want to draw your attention to is where you see these big spikes. So for example, let me take this particular gene right here, which is KRAS. Now you can see for some cancers, there's nothing going on. There's very little mutation in that particular gene. Other cancers, there's very significant mutations in it. Okay, so these cancers really differ greatly in terms of what is responsible for the cancer itself and what the best treatment strategy may be. And again, we can look at any of these other genes, and you can see in certain cases, there'll be a spike for one cancer maybe, and that's it. So it allows us to, first of all, look at these different cancers and understand what is motivating them, if you will, what's causing them to be the disease they are. But as you can imagine, it's also a little frustrating because it makes us realize how complex cancer is, and that one treatment strategy is certainly not going to fit all cancers. Now, this gets a little bit more complicated because even within cancers themselves, there are subclasses. So let's look at this particular type of lung cancer. And all we're going to look at is certain mutations and how predominant they are in this lung cancer. Well, in 23%, there's a mutation in this gene KRAS. In 15%, it's in EGFR. There's no mutation in 56%. So right away, we have subclasses within this one type of cancer. And that tells us something about how to treat them. But again, it does make it a little more complicated. Now we have classes within cancers. And if we drop down to melanoma, we can see the same thing, where if we look at certain mutations in one gene, it shows up 39% of the time, another gene 18, no mutation 35. And even if we get to one of the particular mutations, one of the genes, so this is BRAF, you can see it's not even the same mutation all of the time. 80% of the time, it is a common one, this valine that is changed to the amino acid glutamate. But 15% of the time, it's this valine changed to lysine. And this can, again, affect the treatment strategies. So this information does help us in terms of understanding the complexity of cancer, but it also makes us realize that there may be very different treatment strategies depending upon what an individual has. What mutation you may have could well determine how we're going to treat you. Okay, but in general, the fight against cancer actually takes place in every cancer patient, and it occurs between the cancer and our immune system. That it happens if the immune system recognizes the cancer as foreign. So the way our immune system works is something enters our bodies, the immune system says, wait, that's not supposed to be here. It then attacks it and can hopefully remove whatever it is now invaded. But it has to recognize it as being something different than what's normally supposed to be there. So first of all, does the immune system recognize cancer cells as being different? Is there something different about them that the immune system can pick up on? So let me just take an example of the sugars that are present on the outside of our cells. So on each of our cells, we have different proteins, and then there are sugars attached to them. And just an example I'm giving here, this is the type of sugars we see attached in normal cells. And it doesn't really matter the different types of sugar. You can just see there's a whole bunch of them in a very specific order that we find on the surface of normal cells. And these are really kind of long strings of sugars. Well, what can often happen on cancer cells is you don't see these long strings of sugars. Now you see very short strings, and the sugars are actually different. If you notice, this purple one shows up here on the cancer cells, but doesn't show up in your normal cells. So that at least tells us that there are some things on the surface of cancer cells that differ from the surface of normal cells. Therefore, immune cells should be able to recognize cancer cells and say, that's something different, that does not belong here. I need to get rid of it. Okay, why doesn't it work so easily? Okay, and this just further emphasizes the point I was just making, where you can see these short chains of sugars versus the very long ones. And as I mentioned, there are a number of these sugars associated with tumor cells that are different than what we see on normal cells. So the immune system, again, it's supposed to distinguish self from non-self, okay? It's supposed to work to eliminate things like infection, get rid of invaders, while also not attacking your own body, not where we have autoimmunity problems. Now, the immune system, you can have an overactive immune response, okay, due to an infection. That can be very bad for a number of reasons. It can induce autoimmunity. It can also induce other reactions in the body that create real problems. You can also have an underactive immune response, and that's no good because now you're really not fighting off what the invasion may be, even though you're pretty safe in terms of autoimmunity. So really what we'd like to do is to bend this curve. We'd like to do things that are going to eliminate cancer, specifically have the immune system recognize cancer, but avoid autoimmunity. So you're not going to attack your own normal cells. Well, let's look at what goes on inside a tumor. All right. Now, A tumor is not sitting there all by itself, okay? So here's a tumor, we have tumor cells all around, but you'll notice there's a whole bunch of other cells in this environment that are all over this tumor. And what these cells are these are different cells of our immune system. Now, our immune system really falls into two categories. One is what we call the innate immune system, and one is the adaptive immune system. And they're complementary. And the idea is that they're supposed to provide a defense against foreign threats. So in theory, these different immune cells should come along, recognize the cancer cell as foreign, and then attack it. Okay, and you can see we have natural killer cells, we have cytotoxic T cells. These are pretty effective at getting rid of invaders. But tumors can use various mechanisms to escape detection of these various cells of the immune system and then enable their growth. So let's talk a little bit about how do they escape. All right, well, first we have to get a little bit into the immune system itself. So I mentioned we have two things. We have the innate immune response and we have the adaptive immune response. Now, innate is the first line of defense. And this is where we have our natural killer cells, okay? And now these cells just don't go around all on their own killing everything they run into. Obviously, they're going to be regulated. They're going to be activated under some circumstances and inhibited under others, okay? So that's a way of controlling the response. Now, our adaptive immune response is a little different. This is a more durable response, but it does have to be activated by the presence of a foreign invader. This is where cytotoxic T cells come into play. And again, just like natural killer cells, they're going to be activated in certain ways, and they're going to be inhibited in certain ways. Because again, we don't want them just acting on normal cells. And often these inhibitory mechanisms are referred to as checkpoints, okay? They keep the immune system under control so it doesn't just run amok. Now let me get into a little bit more detail. Don't worry about all the text here. I'm just going to talk about the way that we control first the innate immune response. So we have our natural killer cell, and it's going to have molecules, different proteins that are going to stick off the surface. And one way they can be activated is that they recognize a molecule on another natural killer cell. But also they may have a molecule, in this case, well, this is a CD137, and a tumor cell may have something on its surface that's recognized by this molecule. So this would be referred to as a receptor, and this is a ligand. So this is one of the ways that natural killer cells may be regulated. They can find ligands that they recognize as being foreign, and they will attack the cell that has the ligand on the surface. On the other hand, they can be somewhat controlled by other natural killer cells. Now, let's look at the adaptive immune system, and something similar goes on here. So we have our T cell, and a T cell has a whole bunch of different proteins on its surface, and they're controlled in different ways. And the way they're controlled are by other antigens, and these antigens may activate the T cell, or they may shut it down, okay? So here we see CD28, CTLA4. Now, CTLA4 is really important because this is actually referred to as a checkpoint receptor. In this case, when it interacts with something, that's going to shut the T cell down, okay? That's going to slow its response. In similar fashion, here's another receptor on a T cell. This is referred to as PD-1. This is also a checkpoint receptor. And again, this interaction is going to slow the T cell down. It's going to tone it down. So as you can imagine, the way tumors can elude the immune response is they can produce these ligands that interact with these checkpoint receptors and essentially fool the T cell. They can say, no, we're not something foreign. Just relax, don't kill us. And that's exactly what happens. So one of the big issues in trying to have the immune response interact with cancer cells is you have to get around these checkpoints. You have to somehow disengage this interaction so the T cell can then respond to other antigens on the tumor cell and kill it. Okay, well, this gets to be very complicated because there are a lot of potential checkpoints. Our bodies are really well set up to not attack itself, okay? And you can see that we have a whole host of different receptors here. But But how can we potentially deal with these checkpoint ones? Okay, so here's our T cell. Here's a whole bunch of different receptors, and these are predominantly checkpoint receptors. Well, let's say we created an antibody that has a high specificity for this particular checkpoint receptor. If we blocked it, then it cannot be fooled by a tumor cell, because the tumor cell, when it expresses the ligand can no longer interact with the receptor anymore because the antibody is blocking that interaction. So this is where the concept of checkpoint inhibitors came from as a way of improving immunotherapy. The idea that you would create an antibody or some other molecule that would block the receptor, whether it be the CTLA4, PD1, VISTA, and then that would prevent the engagement of this receptor with the tumor cell, so it wouldn't calm the tumor cell, it wouldn't calm the T cell down. The T cell could then go ahead and destroy the tumor cell. Okay, so let's look at this a little more specifically. Again, here we have an antibody that's been created to block this particular checkpoint receptor, the CTLA4. What's going to happen is that then an antigen can be presented. The T cell will say, okay, there's something foreign here. It will become activated. And it's not going to be toned down by the checkpoint receptor. Okay, so how well did this work? So this is probably the first data that was presented, certainly the most outstanding data, and really moved this whole approach along. Now, graph's a little bit complicated, but let me just point out what's really going on here. So this is a combination treatment in advanced melanoma. We're looking at blocking 2 checkpoint receptors, okay, so that PD1 is being blocked and the CTLA4 is being blocked, all right? So we've got 2 different antibodies. They're blocking 2 of those checkpoint inhibitors. So what happened is that in these melanoma patients, and this is advanced melanoma, there was an overall response rate of 60%. That means 60% of the patients showed a response to this approach. There was a complete response rate of 25%. And ultimately what that meant is that the median survival was greater than 40 months, where the expected survival for these patients was about 7 months. And many of these patients within this 25% group were, you don't like to use the term cured of the disease, but the disease was in complete remission. Now, this was outstanding because typically for this patient population, this is advanced melanoma, again, survival is very short. And what you normally look at in this group is extending survival by several months by different treatments, where here you had virtual complete remission for 25% of the patients. And this is what, as I said, really moved this idea of using these checkpoint inhibitors forward for immunotherapy, for having our own immune system respond to the cancer. Now, of course, this leads to quite a bit of follow-up questions. Response rate is great for 25% of the patients. Why did this not happen for all the patients? There was a certain response rate in a majority, but not the complete response. So we need to understand a little bit more about the immune response to different cancers. Okay, but I'm sorry, I got one slide ahead of myself. So first we had this introduction for melanoma. We then had approval of the 2 antibody approach for melanoma. Then we started to see these checkpoint inhibitors benefit for other cancers. So in addition to melanoma, it's using in head and neck cancer, lung cancer, kidney cancer, bladder cancer, Hodgkin lymphoma. So, you know, this approach now has a great variety of use in different cancers. And again, the effectiveness, it's been similar in different cancers where you see a certain group within that, the patient population that does very, very well. But there's still the mystery why others weren't doing so well. Okay, so how do we improve immunotherapy? Well, it turns out that, at least for some of these cancers, the immunotherapy works best for tumors that are very heterogeneous and have many mutations within the tumor. Okay, well, this is a little bit of a problem because you can't go and introduce more mutations into a tumor. That's not an effective strategy. So what are you going to do? How do you make the immunotherapy better? Well, first of all Are there other checkpoints that could be modulated? And it turns out that yes, one of them, VISTA, was actually a little bit more predominant in pancreatic cancer. So this required a more discovery about what are the actual checkpoints that are being engaged in different cancers. The microbiome influences the effectiveness in immunotherapy, and I think this is going to be a really substantial area of breakthrough over the next decade. So what we have in our gut, the different bacteria that live there, produce all types of molecules. And what we've been finding is that based on the types of bacteria people have, immunotherapy can be more or less effective. Now, recently there was a paper describing a molecule produced by certain bacteria called inosine. And it turns out that inosine helps in the immunotherapy approach. So now we're going to be looking for, in people who are undergoing immunotherapy, do they have certain bacteria in their gut that's producing this inosine, or can we introduce it? So this is one approach that I think is, like I said, is going to be very important moving forward. We can also combine other approaches with immunotherapy, such as chemotherapy, and I'm going to talk about how that works in just a moment. And also we can look at these enzymes that actually remove some of the molecules from the surface of tumor cells that are recognized by natural killer cells and T cells and inhibit those enzymes. So one of the other things that goes on is tumor cells produce these different antigens, but then they're removed off the surface, and that prevents the natural killer cells or the T cells from recognizing the tumor cell. So I'll spend a few minutes and I'm going to talk about these two approaches. Okay, so this is a study that was done where this was just looked at using a typical chemotherapy, using platinum-based compounds, and then using one of the checkpoint inhibitor antibodies. And what you can see is that looking at tumor volume, that just the chemotherapy alone really wasn't very effective. Using the inhibitor checkpoint seemed to work for a while, but then its effectiveness seemed to wear off. But if you combine the two, you had excellent effectiveness and long-lasting effectiveness. All right, why did this work? Why did introducing chemotherapy help in terms of then the checkpoint inhibitor? Well, what it really did is it created inflammation in the tumor. So the chemotherapy treatment was just creating a more inflamed tumor. Well, that more inflamed tumor was more susceptible to the immune response once you got the checkpoint inhibitor engaged. So you had your chemotherapy occurring over a certain time period, and that would create your inflamed tumor. Once you got to that inflammation point, you could then go in with your immunotherapy, which would then be much more effective, and now the inflammation of the tumor would slowly decrease, but you had your effectiveness in reducing the tumor greatly. So this is really a nice example of where you can combine two approaches to get the best possible response in a treatment. Okay, now let me talk about enzymes that sit on the surface of tumor cells and can create some real problems. So this is just an example of a tumor cell, and there's just a lot of things going on here, but what I really want to point out is that if you look at the surface of the tumor cell, there's a whole bunch of different proteins and other molecules that sit on the surface. So one of these proteins is an enzyme, and this is an enzyme that my lab studies, and it's called membrane type 1 matrix metalloproteinase, or MT1-MMP for short. This enzyme does a number of different things to actually aid a tumor cell in growth and also invasion. But what's important about this enzyme is it's a protease. That means it cleaves other proteins. And you can see an example here of where it's cleaving this molecule off the surface of the tumor cell. And this is something called CD44. So when MT1-MMP sits on a tumor cell surface, it has the ability to cleave off a number of different molecules. So it's knocking proteins off the tumor cell surface. Well, how does that impact immunotherapy? So if we go to our natural killer cell, what we can see is that there are certain molecules on the tumor cell surface that it recognizes. I want to point out this particular ligand, which is under this family, which is this NKG2D ligand right here. There's a member in that family called MICA. Well, it turns out MT1-MMP removes MICA from tumor cell surfaces. So normally a natural killer cell would recognize the tumor cell by the production of this MIC-A, but the enzyme is removing it, and that's going to slow down the ability of the natural killer cell to recognize the tumor cell. So this is another area that not only our group, others are working on. These enzymes produced by tumor cells that help disguise the tumor cell, how to inhibit those enzymes, block their action, so then these different molecules stay on the surface and can be recognized by the immune system. All right, so now I'm going to step a little bit forward in terms of immunotherapy approaches and talk about targeting approaches. Up to this point, I've really just discussed immunotherapy as far as our natural immune system, but now we're going to look at some ways in which we can utilize some more targeted approaches. So the first thing I'm going to talk about is something called CAR T cell therapy, which, again, some of you may have heard of. In this particular case, you're taking T cells from a patient. You are reprogramming these T cells to find and attack cancer cells, and I'll discuss how that's done in just a moment. You then grow up these T cells in the lab, and you put them back in the patient, okay? So this sounds pretty straightforward. How are you reprogramming these T cells, though, to attack a tumor? Now, normally, if we look at the structure of an antibody, okay? So antibodies are highly specific, or usually highly specific, and they recognize very specific antigens. They have certain molecules they go after. Now, you do have antibody-like receptors sitting on B cells, and these will recognize, again, certain antigens very specifically. So if we can identify antigens from cancer that's in a patient's body, in theory, an antibody would be very specific at recognizing that antigen. Well, what if we could take this antibody and then engineer it onto a T cell? So we take part of the T cell receptor that normally exists, but now put this antibody-like molecule here. So we enhance the ability of the T cell to recognize the tumor cell. It's going to recognize a very specific antigen. And that's really what CAR T cells are about. So the CAR is the chimeric antigen receptor. So we have our normal T cell and the T cell receptor, but now we've engineered it through a viral vector transfection, where it's going to produce an antibody-like molecule on the surface. It will recognize an antigen on the tumor cell, and that will allow it to very specifically attack the tumor cell. So we've re-engineered the T cell to be more selective in terms of what it will now focus on. And here's sorry, this is a little bit blurry, but here's an example of the way this is done. So normally in a T cell, you would have this receptor, this CD3, but now we're going to take part of this antibody, this CD20 antibody, and engineer it onto the surface of the receptor. So now this is the receptor in the T cell. It's a chimera. It has this part of the normal T cell receptor, but now this antibody engineered CD20 part. Well, the tumor cell, there's a subset of the tumor cell that highly expresses the antigen for CD20. The T cell is going to interact with it. It's going to eventually kill the tumor cell. The question here is more going to be how well will it penetrate into the tumor, but hopefully in the end, maybe you end up with tumor eradication. At the very least, you have a pretty effective killing strategy. And this is just one example of some work that was done on large cell lymphomas. So this is using CD19 recognition in the CAR T cell approach. And you can see the before treatment, about 2 years after treatment. You can see here was some very extensive large cell lymphomas. And again, 9 months after treatment, it's been greatly reduced. And then here we have another example. And again, 5 months after treatment, looks like it did pretty well. So this is a very promising targeted approach that is now being used. It's an immunotherapy approach, but it's now targeted. Now, like all approaches, there are things that happen that are problematic. So there are complications of CAR T cells. There's what's called cytokine release syndrome. Occurs usually within 5 days. there's a rapid T cell proliferation, and that leads to the release of different cytokines. And that, unfortunately, can lead to shock and subsequent organ failure. But it's not a total dead end there. This can be treated with monoclonal antibodies against interleukin-6, and also can be treated with steroids. So there are some issues, but this is certainly a very promising approach as we move forward. Now, while we're still talking about targeting, let's talk about a really simple targeting approach. So instead of putting an antibody on the surface of a T cell, let's just take an antibody and hook a drug to it. So this way, the antibody will be recognized by, or will recognize the ligand on the surface of the tumor cell. It all gets brought into the tumor cell by what's referred to as receptor-mediated endocytosis. Once it's inside the tumor cell, it then will interact with the lysosome. The lysosome has a bunch of enzymes, proteases, that chew all that up. Once it chews it up, it releases the drug. And once the drug's released, it can go to the nucleus, if you will, of the tumor cell, and that will kill the tumor cell. So this is the idea for antibody-drug conjugates. All right, so this just sort of summarizes a few approaches I've discussed that are all really relate to using immunology. We talked pretty extensively about these checkpoint inhibitors, using these antibodies as a way of allowing the T cell to be more active against tumor cells. We talked about these CAR T cell approaches. Okay. We talked about the antibody-drug conjugates. Okay. Now, as you can imagine, there are things I didn't discuss here. There's radioimmunotherapy, where now you have hooked to the antibody rather than a drug. You've hooked a radioisotope, which can kill the tumor cell, and so on. There are other approaches too that work here in terms of using immunotherapy. So let me go back to my pillars. Again, we had the ones that are pretty well known: chemo, surgery, and radiation. We've talked about some targeted approaches here, and we've also talked about the new advances in immunotherapy. I'd like to finish with just a couple of slides, though, talking about radiation therapy, because radiation therapy for a long time has been thought to be just one type of approach. It really isn't, and there are some really exciting new advances there too. So what I'd like to just bring up is something called carbon ion radiotherapy. So in terms of carbon ion radiotherapy, why this is such a promising treatment is if we look at X-ray treatment of cancer, X-rays have a certain penetration through the body to get to where cancer may occur. And then their ability to really do damage to the cancer, it drops off pretty dramatically. What is very interesting about carbon ion is that it works almost even better when we get to certain depths in the body. Works very well. And this can be important for certain hard-to-treat cancers, things like lung cancer, pancreatic cancer, certain head and neck cancers. Now, why is this so effective? Well, carbon is really, it's a pretty big molecule, especially when we compare to things like X-rays or even protons. Now, some of you may be familiar with the South Florida Proton Therapy Institute, which is in Delray, and that uses protons to treat different cancers. Well, carbon is more effective because it's a larger molecule, and when it gets to the cancer, it can do significantly more damage. Well, how does this really work? So this is the setup for doing carbon ion radiotherapy. You have your ion source, you're going to have an accelerator, but most important is you're able to direct these beams very specifically at the patient. Okay, so this is not just random radiation floating around. It's very, very targeted. You can do this vertically, you can do this horizontally. You can actually have patients in certain beds where the bed rotates, so you can obtain the best possible angles at doing the carbon beams. It's really very, very precise, and I think is extremely promising moving forward in terms of radiotherapy. Now here's an example for lung cancer treatment, which again, as many people know, lung cancer is not, the treatments are very limited. So here is a PET image before. You can see this fairly substantial tumor here. And here's after. Basically, the tumor has been eradicated. It's gone. So very promising. Now, the downsides, we don't have this in the United States. Right now, all the carbon ion beam treatment facilities are in Europe, Japan, and Korea. But there is movement to bring it to the US. Certainly, there's been a lot of testing going on in Texas for this. The Mayo Clinic up in Jacksonville has talked about bringing carbon ion beam radiotherapy there sometime within a 4-year window. So I'm hopeful that we will see this in the United States sometime soon because, again, I think this is really a great advance in radiation therapy. So that's it for what I wanted to present today. I will be more than happy to answer questions now.
Great, thanks so much, Greg. Just as a reminder, if you have any questions, please hover your mouse on the bottom of your screen. There is a Q&A button that you can push, and please type your questions there. I'm going to start out with the first question, Greg. The microbiome, you talked a little bit about that. And the question here is that the microbiome may change in oncology patients as they progress in treatment, seeing that oncology patients often have a high risk for malnutrition, weight loss, inadequate oral intake, and so on. How does it affect the microbiome?
Oh, and this is an extremely important point because you can go from having a healthy microbiome, which might really aid in your immunotherapy, to having one that does not really help anymore because of the other treatment strategies. So as we're becoming more aware of what's in the microbiome and how it can contribute, I think what we would see is supplemental therapies that would go along, for example, with immunotherapy. So first of all, if there are certain compounds that we recognize the microbiome produces that are helpful, we could certainly just have those compounds part of a treatment strategy. Also, I think we are going to really look at the diet and how we can try to modulate the diet of cancer patients as they're being treated to make sure that they do maintain, the constituents we want them to maintain.
Any evidence that exercise affects the microbiome and or the treatment of cancer patients?
So I don't know exercise in terms of the microbiome, but exercise is certainly a positive for cancer patients. There's no question about it. And it's hard to get into all the details as to why that is. And certainly for cancer patients, that may be one of the most difficult things to do. But overall, it's very positive. It certainly helps to keep the immune system as strong as possible, and in other ways too.
You mentioned immunotherapy for a number of different types of cancers, but certainly not all. Is it your expectation that these cancers that you're basically missing right now on your list there, they would benefit as well from immunotherapy down the road, or is it truly limited to certain types of cancers?
So let me take one particular cancer, for example, which is pancreatic cancer. So pancreatic cancer, again, is unfortunately one of those that has a very low rate of survival. And part of the problem with pancreatic cancer is that when it occurs, the cancer itself sort of forms a nest around the cancer. It's what's called a fibrotic nest, very dense in collagen. So it's protecting itself. And immunotherapy hasn't been great in pancreatic cancer yet. So one of the strategies people are looking at is, well, how do you deal with that fibrotic nest? Can you degrade it? Can you have other treatments that will open it up some that then allow other approaches to work more effectively? So I think in those type of cases, we're looking at combination therapies. Will it be a chemo plus immuno? Will it be a radiation plus immuno? I don't know right now, but I think that's where we're going to get to on some of these cancers where the immune system just isn't able to do much. Now, brain cancer is a totally different story. Okay, so our blood-brain barrier is set up to not allow the immune system to start invading in there. So there we have to think about some different strategies.
Great, very interesting. Which kind of leads to our next question here. How do doctors decide which of the myriad of biological mechanisms to target in cancer treatment?
Right. So right now there's so much information out there about what I mentioned earlier about these different mutations people have. That certainly will help guide certain types of chemotherapy, no question about it. I think it's very important that there is a genetic profile for a patient when one considers treatment strategies. Immunotherapy, it does depend on the type of cancer they may have. I talked about melanoma as an example, advanced melanoma. Certainly there can be, in the genetic analysis, you're going to be able to tell how much mutation has occurred, whether you have a better feel for if immunotherapy is going to be very effective there or not. So a lot of this comes down to the gene profiling of the patient and understanding what are the mutations that are present and what are the treatment strategies that best work based on those type of mutations or just general number of mutations.
So is determining the gene profile of a patient standard of care at this point?
I would hope so. I'm not out there with every physician. We're doing what we can in terms of trying to bring that forward from the standpoint of research and analysis. But I don't know how many doctors utilize that approach, how many don't. But certainly it's made significant progress, and you do see a lot out there.
Great. We have a question related to BRCA genes, and where the T cells do not function properly. Would immunotherapy be a possibility with those type of cancers?
Well, so ultimately the answer is yes. It's just a matter of which type of immunotherapy and how you can work with different cancers. For example, let's take the CAR T cell strategy, okay? So if you're able to take cells, grow them up outside the body, right? Get a very specific targeting function in them, that can help override if your normal immune system might not be functioning particularly well, okay? So, things like, this is also a place where something like the antibody-drug conjugate could be very important, right? So, now you're just using antibodies directly. You're not relying on T cell function at all. So again, it's consideration of how severe, a certain problem is. If you can't override it one way, do you start to go to more of a targeted approach where you can use antibodies?
Our next question is related to computational systems. Which, if any, computational systems are in use that help doctors decide the most or the best course of cancer treatment?
Oh, that one I can't say I have an answer to. There are certainly approaches where you can look at what someone's gene profile is and use various, artificial intelligence, so on, to determine what would be the best outcome course, what drugs might be effective, what wouldn't, which ones may cause the most side effects, and so on. But I can't actually say which particular program software doctors may be using. It's maybe very dependent on the hospital and so on.
Mm-hmm. What about prognosis? A lot of what you mentioned it earlier. A lot of the best treatment for cancer, and then certainly surgery, depends on how early cancer is being detected. Are there any major advances in prognosis of cancer that you can talk about? Right.
So the different imaging has certainly led us a long way. And that's quite important. Right now, you see a lot of research devoted to biomarkers, okay? Are there things circulating in the blood that might give us some advance notice? Now, again, I'll go back to pancreatic cancer, which has typically been a very hard cancer to diagnose early because often there aren't symptoms. By the time there are, it's already spread a bit. That's a cancer where they're looking more into, well, what's circulating in the body in advance? Are there some things that could let us know well before we detect this through other ways? And that's where biomarkers in the blood come into play. So that's certainly been an area of great advance and extensive research.
You mentioned earlier the heterogeneity or the diversity, the differences in one tumor. When you have one tumor from one side to the other, you may have a different profile. How does that affect the success of treatment? And how does the genetic profile address that heterogeneity?
So that's really, first of all, it makes things much more problematic in terms of treatment. When you talk about genetic profile, yes, are you getting an average of what's going on in a whole tumor, or can you get down to specific cells? So certainly an approach that's been very prominent the last several years is single cell sequencing, approaches where you can look at profiles in individual cells that can potentially allow you to see how much heterogeneity may be present rather than getting some average across the tumor. But it does make things very problematic. And one of the real issues in cancer, especially when we get to the stage where it's metastatic, is often you see a reduction in a tumor, but the cancer comes back. And part of that is the inability to treat certain subsets within the tumor. And that's what we're really trying to understand. How can you effectively develop strategies that might treat all subsets? Or after you've effectively knocked out one subset, you can then go after another. And but no, this is clearly a problem. And this is why the term remission is used rather than cure, because cancer goes into remission, but there is some subset that may still be unaffected and is waiting to sort of spring up again at a later time.
Yeah. Given that the best way probably is to avoid cancer is trying to prevent cancer, do you have any suggestions on how to best prevent cancer? I mean, it's probably common knowledge at this point that don't smoke.
Right.
But beyond that, do you have any suggestions?
So, certainly one of the correlations we're finding over time are certain diseases, as they become more prominent, then cancer becomes more prominent. So something like inflammation, all right? And now we're starting to understand more and more why inflammation, chronic inflammation, can then eventually lead to cancer. So one thing to do is try to avoid chronic inflammation. How do you do that? Well, that's where something like exercise can come into play, by making sure, trying to make sure you're in as good a shape as you possibly, can be in, to avoid things that are then going to cause injury that will eventually lead to inflammation and so on. So I think, part of it is learning, how exercise comes into play, and certainly part of it is diet. And, we hear, I don't know, to some degree, I think people are maybe hear too much about diet, but it becomes very, very important, because all these things are interrelated, right? Diet is going to affect your ability to then, be relatively healthy. It's going to affect your ability to exercise. These things all have to come into play. So beyond, certainly, not smoking is at the top of the list, but, just doing things that will avoid inflammation, doing things, trying to exercise, trying to be careful in terms of diet and so on. Yeah.
Speaking of diet, is there any evidence that alcohol affects cancer in any way, or cancer?
So that's been all over the map. I mean, for the most part, I think it's generally agreed that limited alcohol intake has its pluses and minuses. So, a glass of red wine with dinner at night is not going to significantly increase your propensity, your possibilities of getting cancer. Excessive alcohol drinking, well, that leads to conditions that can eventually lead to cancer. Certainly, the more you tax your liver, that's problematic. So, but like anything, we have to look at moderation versus excess, okay? I mean, sugar is another, can be a very bad actor out there. Now, some sugar we need, you need it for brain function and for a whole bunch of other things. So, you don't eliminate sugar from your diet. But, we are aware of the fact that, having too much sugar puts stress on certain parts of your body. And again, ultimate outcomes can be very poor. So you want to moderate that. So yeah, there are certain things one can look at as far as intake and say a little is fine, a lot is not fine.
Great. We're about out of time, and the last question is one you may want to answer in an email or something, because it's related to publications that address the microbiome. So that
Okay.
we're probably going to address that offline. So
Okay.
with that, I want to thank you, Gregg. Very interesting. And we hope to see everybody on the call today again next week, next Thursday at 1 p.m. Thanks so much. Bye-bye.
Right.